going to talk now about the microbiome, which is a revolution in itself. And we use the word revolution a lot, but honestly, and I've been through a lot of them, there's nothing quite like this. This famous picture from the moon, from Apollo, of the Earth as a water planet. It turns out that almost all life, by many orders of magnitude, on this planet is in fact microbial. It's everywhere. And you know, I started as an astronomer, astrophysicist for 30 years. You know, I did it because I wanted to go into something that was complex. There were 100 billion stars in every galaxy, like the Milky Way or Andromeda, and there are 100 billion, now they say there's maybe 1 trillion galaxies, each with 100 billion stars. That's astronomical, right? But on this planet, there are 100 million times as many bacterial cells as there are all of the stars in the universe. And yet we know very little about it. And by the way, that includes inside and on of all of you. So this is a great quote I love, which you'll notice I'm no longer doing astrophysics, I'm now doing microbiome, and it's because the complexity inside each of you, the universe inside, is way more complex than what goes on in astrophysics. And we're going to basically figure out how to map that universe. And so Rob, my colleague at UC San Diego, is a co-founder of the EarthMicrobiome Project, and he's going to take you a little bit through where we are, and this is really up to date, as he'll tell you, being published just last week.
That's right. Thanks, Larry. So what we've been doing in the Earth Microbiome Project is mapping microbes on scales from our bodies to the entire planet. And the paper we published just last week describes the first 27,000 samples crowdsourced from around the scientific community, although we're over 100,000 samples in the project now. And this was initiated with government resources at the DOE at Argonne National Lab. But when Jack Gilbert and Janet Jansen and I founded it back in 2010, we initially thought it'd be a large-scale government infrastructure project, but ultimately it was primarily picked up by private foundations and in-kind contributions from various companies, which allowed us to get a start on it. So where it dates back to is a decade ago when I was working with one of my very talented grad students, Kathy Lozapone, and she was interested in what could we do using the universal tree of life as a measuring stick to compare microbes from anywhere on the planet. So we went to GenBank, the public database that has all the DNA sequences, and we got sequences from all over Earth, so everywhere from the poles to the equator, extremes of temperature, pH, just about any other biotic or abiotic factor you could imagine. And the question was, out of all this complexity for microbiomes all over the planet, would there be any clear pattern that could let us describe them? And astonishingly to me, the answer was yes. So you see this very clear split, and so this was back in 2007. Just to give you an idea of the scale of data increase, we had about 200 samples and about 20,000 sequences, which was a massive data set at the time. And so much to my surprise, out of all this complexity emerged this very clear pattern. So you see this very clear split between saline samples in green and non-saline samples in red. And you see a few blue symbols, which are mixtures between the saline and non-saline, so things like estuaries and things. So out of all this complexity, we see a clear pattern. And it wasn't the one we expected, because you might have expected, say, temperature to be the main factor driving microbiomes on Earth. But what we see when we look at high-temperature environments like hot springs and hydrothermal vents is they just look like the other non-saline and saline communities, respectively. However, in later work with Jeff Gordon's lab, we were able to search really hard for environments that truly were extreme from a microbial perspective. And you might wonder, well, how far do you have to go to find these extreme environments? Do you have to go to the top of a mountain, the bottom of an ocean, or what? And amazingly, you don't have to go far at all, because they're right there within us. And when we add these environments, mostly from the mammalian gut, we get this new axis on this plot that explains twice as much of the variation as that saline, non-saline split I showed you. And what we're looking at is primarily samples from humans and mice. Remember, this was a decade ago. That's all we had access to then. And it shows that the mammalian gut is profoundly different from any kind of environment that you see out there in the oceans and the soils anywhere else on the planet. And we're still trying to understand that. So what happened in that last 10 years? Well, what happened was that we went from this 200 samples, 20,000 sequences, to this just a decade later, 27,000 samples and 2.2 billion sequences, which is what we described in the recent paper. And one thing that was, and so this came out just last week, where essentially with about 400 co-authors, we were able to provide the first delineation of Earth's microbial life all over the planet. And one of the key contributions of this paper was this, the EarthMicrobiome Project ontology, where instead of taking our preconceived notions about what mattered from a microbiome perspective in different habitats, we let the data guide those decisions, where you see the deepest split is between free-living and host-associated microbes, then within the free-living between saline and non-saline, and so forth at finer and finer scales. And we mapped all these different categories all over the planet with those 27,000 samples. And so this gives us the first overview of how microbial life is arranged on Earth. So that's all very well, you're thinking, but how does this relate to medicine? And in many ways, what's inside is really what's outside, right? Your body is fundamentally a torus with this tube running through it, and the microbes in your gut are feeding off materials that are coming in straight from the environment. And in the Human Microbiome Project, which was a large-scale NIH-supported project that I was involved in several capacities, we basically took this concept of the body as an ecology and looked at microbes from all over the body, looking at the different kinds of microbial life in different locations. We also complemented this with crowdsourcing and crowdfunding. So as you probably know, most crowdsourced citizen science projects are on the scale of a few $1,000s. The American Gut Project, which we launched in 2012 over Thanksgiving, Thanksgiving being a time when a lot of Americans are thinking about their gut for some reason, we launched this project to essentially open these techniques up to anyone who wanted to participate through crowdsourcing and crowdfunding, so that it's kind of like National Geographic's Genographic project, where you can add yourself to the project just by covering the cost of processing your sample. And so we've raised over $2 billion. We've sent out 12,000 kits, of which 10,000 samples have already been sequenced and released free to anyone who's interested in looking at them. So what do you get out of this sort of microbiome mapping exercise? Well, what you're seeing is based on technology we developed in my lab, so looking at the unifrac distance metric for comparing microbiomes using Darwin's fundamental insight that there's one tree of life that you can connect all organisms to, so you can use that tree as a measuring stick to compare communities. And what we do is for each community, we read it out from the DNA of its microbiome, and we place it onto this map where 2 points are close together if they have more similar microbes in terms of their shared evolution, and they're further apart if they have more dissimilar microbes. So all the complexity of this oral sample that I'm showing you here is being read out from its DNA and distilled just down to 1 point on that map. And so the main thing going on becomes immediately clear. So remember, these are about 250 healthy people. We're not seeing any differences in disease state. So what we see is primarily that the different parts of the body are totally different from one another in terms of their microbiome. So you see the mouth in green, the skin in blue, the vaginal samples in pink, and the stool samples down the bottom in brown. And what's amazing is that these different sites of the body have completely different microbiomes from one another. And it wasn't until we did the EarthMicrobiome Project that we fully started to begin to appreciate these differences in the microbiome, even within ourselves. And to illustrate this, I'm showing you the mouth and the gut from the first person in the Human Microbiome Project. And so when we cross-reference the information to what we saw in the EarthMicrobiome Project, looking at different kinds of habitats all over the planet, we could finally ask what two sites on Earth are just as far apart in terms of their microbiome as the mouth and the gut of this one individual. So if you think about your mouth as being kind of like a coral reef, it's covered with complex mineralized structures that maybe your dentist pesters you about. The amazing fact is that your mouth is as far from your gut in terms of its microbiome as that reef I'm showing you is from that prairie. Right? They're completely different kinds of microbial life and microbial organization, which is pretty remarkable when you're talking about two ends of the same tube. Right? It means a few feet along the length of your body makes as much of a difference to your microbiomes as thousands of miles across the Earth's surface. And we're just starting to understand how each body is made up of an archipelago of different microbiomes and how they're communicating with one another. So one really important part of all this is trying to understand what is the impact on our internal microbiome from environmental influences. And one of the most important environmental influences is the food that we eat. So what we're finding is that diet has one of the largest impacts that we've seen on the microbiome of all the different things that affect it. And if you want to make yourself look like a different person, a really good way to do that is to change what kind of food you eat. But you can't do it overnight, right? Like, it doesn't matter what you happened to eat at dinner yesterday, but what you ate over the last 6 months and what you will eat over the next 6 months has a profound influence on your microbiome, and is what's going to change it to look like the microbiome of a different person. Anyway, to answer this question in more detail, together with Peter Dorrestein and Rachel Dutton, two very talented faculty members at UC San Diego, we're launching this thing called the Global Foodomics Project. And at the moment, we're still acquiring initial data for this. So what we've been doing is we've been running thousands of food samples as they're about to be consumed. So the food as it's prepared, not just the raw ingredients, through a mass spectrometer, which is basically a kind of molecular scale that weighs each molecule very precisely. And we smash those molecules up into tiny fragments that allows us to piece together, almost like a puzzle, what the original molecule was. But the thing is that most of these molecules are unknown. So in large part, we have to do it by matching up these fragmentation patterns to the molecules that we already know. And essentially the goal here is to take the Global Foodomics Project to understand what's going in, the American Gut Project to understand what's going out, and then to do meta-analyses that connect the two so that we can integrate all this information to understand what are the effects of food on health, and is the effect of your dietary ingredients greater or less than other things that you might be doing, like vitamins or probiotics, or even things like antibiotics and other medical treatments that affect the microbiome. So what we get out of this is this kind of very complex picture, and I'm not going to go into too much detail. I'm just going to tell you that each node on this graph represents a particular molecule, and it's colored according to what the food source was. So red for grain, blue for dairy, green for vegetables, and so on. And what we can do is we can take all those molecular fragments and piece them together into this puzzle, so we can ask what kinds of molecules are in what kinds of foods in an unbiased way for the first time. And what we're getting out of this is amazing. So we have a combination of things that we expect. So for example, what you're seeing here is just zooming in on a little bit of that molecular network, looking at piperine, which comes out of peppers and other related natural products. But we also see a lot of things that we don't expect. So for example, when you take a lemon and cut it up into the lemon peel versus the inside of the lemon, we start to detect a pesticide. So here you're looking at thiabendazole and one of its derivatives. And in general, we're finding a lot of substances in foods that we don't expect. And in part, this was motivated by the American Gut Project, where we asked people about many things, including antibiotics usage. And for example, a fairly large fraction of the American gut population swear they haven't been on antibiotics for the last year, but when we run their fecal sample through the mass spec, sure enough, we find evidence that they've been on antibiotics very recently. So we started asking why, and a really big problem at the moment is just the problem of fake food and lack of provenance in the food chain. So you might have seen reports that, for example, in LA, half of the sushi that you buy may be the wrong species, not only coming from the wrong aquaculture setup, but the wrong species entirely. And similarly, there's increasing evidence that shrimp are getting very high doses in factory farms, especially in Southeast Asia, including antibiotics that are not approved for use in humans, where you can get therapeutic doses of those antibiotics by eating the wrong shrimp. And we're now able to read that out both from the food and from what comes out the other end of our subjects. So another thing that's really exciting is with this common reference frame, everything from different parts of your body to different parts of the entire planet, we're now poised not to just take these snapshots of the microbiome, but to study the microbiome over time and discover the impact on the microbiome from common medical interventions. And so, for example, when people find out about the American Gut Project, they tend to be a bit skeptical about, do you really have all these people sending in fecal material to your lab? And so I tend to get a fair amount of crap from my colleagues at UCSD on that topic. And in this one particular case, it was literally true. And so this box from Larry showed up in my lab on dry ice. And Larry's going to tell you a little bit about his unique story with respect to that box.
Well, it's been fabulous having Rob come to UC San Diego. He's been there for years now and built up a huge lab, one of the best in the country. So as a result, I am able to now do detailed time series. Like for instance, I'm sure many of you, like I did this morning, took a stool sample and froze it. And I'm doing that for 100 days straight while I changed diets as part of an actual clinical trial to look at caloric feeding restriction to just 10 hours a day and what that does. Think about it. These bugs are able to reproduce every few hours, and you're eating, all day long. And the food stuff, you think of it as food. The bugs think of it as a set of chemicals, fibers, and so forth. And they're each little optimized chemical refineries that are dealing with that and producing byproducts. So for instance, the reason that you're supposed to eat your vegetables is because of the fiber. The fiber, the bugs ferment, and one of the byproducts are called short-chain fatty acids, and one of them happens to be butyrate, which is the most important energy source for the human epithelial cells lining your colon. And that's developed over, millions, tens of millions, hundreds of millions of years of coevolution. So, in other words, the bugs do things for us, give us ecological services that we have given up in our own human DNA. Now, I'm a computer scientist. I ran supercomputer centers. I'm a big data guy. What was a profound realization for me was that a single gram of your stool, about 40% by dry weight of your stool, is actually microbes. And they're each, millions of bases long. And so a billion times million, you have basically in 1 teaspoon of stool the equivalent of a ton of DVDs in terms of information content. So it's the richest information material that you ever lay eyes on. Now, naturally, as a computer scientist, I found this wonderful Library of Alexandria. I read the information, right? And what do you do with yours? You flush it? You don't, would you flush, Moby Dick? Would you flush, David Copperfield down the toilet without reading it? So there needs to be a little attitude adjustment about stool. Anyway, what as you heard last night, I found that I had an autoimmune disease, inflammatory bowel disease. As a result, I've had colonoscopies virtually every year, MRIs we talked about last night. And I always wondered, when you drink that gallon of horrible fluid and clean yourself out, what does that do to the poor microbiome? So I was having one in 2015 and said, well, as long as I'm going to go through it anyway, why don't I take stool samples, both going up to it, these are all days along the bottom, and see what happens. And so because we took the stool samples, put it to Rob's lab, it was sequenced, and then we use, by the way, Rob and I currently, a lot of computer time to analyze this stuff. So a lot is like if you ran your laptop for, an hour and then run it for 24 hours and then run it for a week and then run it for a month and then run it for a year and now run it for 100 years. That's how much Rob and I use on the Supercomputer Center at San Diego every year. So just to analyze this data and that of lots of other samples. So here's the diversity of the microbiome in my gut. And that vertical line is when I have the colonoscopy cleanse. And you can see that there's a major reduction in the diversity. And then over weeks, it comes back. And if you look at it in this diagram, this map that Rob's been showing you, where you project the entire ecology down to a single point, the blue dots are the pre-colonoscopy. And then you see, of course, that very large run across the screen. That's between the day before and the day after the colonoscopy. So it sends it very far away from what the ecology was. And then there's this sort of wandering. And after several weeks, you get back to where you were. And that's why generally it's considered a safe thing to do. But if you look in the literature, of course people have done this before, you'll say, well, we took a sample, just after the cleanse, and then we went back 2 weeks later and a month later, and we found about 3x different in say, genuses. Well, here, what we find is because of the higher time resolution, we see there's 40-fold reduction in some genus, and as much as an order of magnitude increase in others, and others just oscillate back and forth. So that means that the whole character of your microbiome is shifting radically as a result of this. Well, last night I was talking about my surgery, which happened about a year ago. And so, of course, I did stool samples through all of that. And these are, again, days. And you see this dramatic shift. First, I had a colonoscopy just literally a week or so before the surgery. And then you notice how the colors abruptly changed. The first stool sample I was able to make with my new colon. That was actually sort of challenging. You know, I had IVs in both arms and an oxygen tank in my nose, and I had IV poles, and nurses all the time, and my legs were strapped to the bed, and I still had to get, you know, the sample and then freeze it, and just had to know when the nurses were hitting in the room and not. So notice that once again, where the colonoscopy, you have this drop. But notice that that's a small, trivial drop compared to what happened after the surgery. And that's 0 at the bottom. So it basically went down to a couple of proteobacteria, was my entire microbiome after the surgery. Now, this is not something that people know about because they haven't bothered to do this at this level of resolution after major surgery. What does that look like in this diagram? Well, these are days. And the orange ones are the days before the colonoscopy. And now look at the colonoscopy. You see you get that runaway and then back to where you were. But now, a few more days until the blue is after the surgery. And guess when the surgery happens? So after that, my colon was resected and then put back together. Took another couple of days to actually be able to work and produce stool, a huge variation. And you say, well, how big was that, say, compared to the diagrams that Rob was showing you about the whole body? But you see, I went from an inflamed colon to one that wasn't. And I did not go back to where I was before. That's the kind of mapping we're trying to do for other medical interventions. And here is that whole diagram. Remember, the pink is vagina. All this is, the brown are stool samples from thousands of individuals. And you'll notice that originally I'm just in one little area, but then I cut across the entire variation. So if you had just one stool sample, and then you had a medical intervention, you have no idea. I mean, I'm transversing everybody's stool samples as far as difference. And that just wasn't really understood before. So you might say, okay, Larry, that's great. You know, you're figuring out that you have different microbiomes. What the hell difference does that make to your later medical evolution as a human?
And so you've seen from Larry the first remarkable journey of someone during surgery that's completely transformed their microbiome so that they look like a new person after surgery than they did before. And by the way, that deviation that you saw immediately after surgery, that represents Larry looking like another species, not just like another person, right? That's an incredible journey through this microbiome space. So having demonstrated that we can change the microbiome in this way by cutting out just the right piece of tissue, we want to motivate you about why you should care about having the right microbiome now that you know you can change it. So one really important thing is that we've been losing a lot of microbial diversity. So with various collaborators, I've been doing studies of groups like the Yanomami in South America, shown here, and the Hadza in Africa. So hunter-gatherer and other cultures living extremely traditional lifestyles. And what's amazing is how different their gut microbiomes, but also their skin microbiomes, their oral microbiomes are from what we see not just in Westerners, but people doing subsistence farming using more modern methods, and other people around the world. And there are entire phyla, entire major groups of bacteria like the spirochetes that primates have co-evolved with, and you'll see those in the gut of people living a hunter-gatherer lifestyle, but you'll never see them in anyone in this room, or indeed if you go to a village in Malawi or in Bangladesh and look at 1,000 people there. So And so this is really important because even large-scale efforts like the Human Microbiome Project have largely focused on healthy Western adults, right? And as soon as you go into other people, other populations, as soon as you go into children, you see completely different configurations of the microbiome that can have a major impact on everything from how you metabolize drugs to various diseases, ranging from things you'd expect like IBS to things that are more surprising like Parkinson's, and even things like how medications will or will not work on you. So a large part of this we think is due to missing microbes, and one thing that's emerging is really important is we need to move away from the war metaphor where all the microbes are bad and you need to eliminate all the bad players, and move much towards managing the human microbiome system as an ecosystem that we need to nurture its health. And I think one graph that really illustrates this very nicely is over the 20th century, through remarkable improvements in public health, single organism pathogen-caused diseases, everything from measles to TB, declined precipitously in frequency. And that was a really important and really amazing advance. But then what you see at the same time is a rise in all these autoimmune diseases like multiple sclerosis, Crohn's disease, type 1 diabetes, asthma, and dozens of others. And so the question is, are we creating a silent spring situation in the gut, where in order to get rid of a few bad players, we're wiping out all kinds of beneficial microbes that are a key part of that ecosystem? And what amazes me is that in 2002, when this article was published in the New England Journal, none of those 4 diseases had been linked to the microbiome, whereas today we know that all 4 of those and dozens of others are linked to the microbiome in humans, and we have detailed mechanistic stories in mice about how exactly they work in an animal model. And so my colleague Marty Blazer at NYU has written this wonderful book called Missing Microbes that goes into far more detail on this. So for public health, we're still thinking about individual pathogens that are bad, but what we need to do is we need to transition that. Instead of thinking about single species, ask more about what is the ecology that permits that bad pathogen to take over the rest of the ecosystem. And one thing that's emerging as especially exciting is the gut-brain axis, where the microbiome mediates a lot of signals between the gut and the brain. And what's fascinating there, a lot of people wonder, how could the gut microbiome affect anything in the rest of the body? But it's important to remember that the gut is not Vegas. What happens in the gut doesn't stay in the gut. And when you're thinking about connection to the brain, everything from signaling through the immune system to the vagus nerve, to the neurotransmitters that Larry mentioned, to hormonal signaling, there's a whole lot of ways that the gut microbes can affect your brain in diseases ranging from anxiety to depression to autism to multiple sclerosis to Parkinson's disease, just to name a few of the stories. And then beyond that, things like obesity, cardiovascular risk, and inflammatory bowel disease, we have very detailed understanding developing of how microbes affect all those conditions. And one thing that's exciting about this, especially in the context of the gut-brain axis, is that there's a lot of studies coming out right now, including many from UC San Diego, that are uncovering these deep relationships between the gut microbiome and various behavioral disorders. And so this covers everything from anxiety and depression, as I mentioned, to autism, to Parkinson's. There's even some suggestions that Alzheimer's and schizophrenia may have microbiome involvements, although that's still very early stage at this point. But what's amazing is how rapidly this has gained common understanding, because when I wrote an article in Dialogues in Clinical Neurosciences back in 2011 proposing that the gut might be linked to different behavioral traits, everyone thought that I was the one who needed a psychiatrist, right? Whereas now we're seeing a lot of mechanistic details about exactly how these things work and how you might be able to change your microbiome in order to improve your health. So again, as you heard from Larry, the microbes that you have in your gut regulate the production of serotonin, but also of other neurotransmitters. And Emran Mayer, one of my colleagues up at UCLA, has this wonderful quote where he says, "It's almost unthinkable that the gut is not playing a critical role in mind states." And what we're seeing is a huge number of very detailed mechanistic studies in mice where we can tell exactly how the behavior works in mice and then start to translate that back into humans. So for example, Elaine Shao, who worked on autism in Sarcos-Mazmanian's lab at Caltech, showed that if you take pregnant female mice and you inject them with double-stranded RNA to simulate a viral attack, their pups develop a whole range of symptoms reminiscent of human autism, where they develop compulsive behavior, so they'll compulsively bury marbles. They They have communications deficits, they have social deficits, they have GI dysfunction. And what's amazing is that a large part of the reason is that the altered microbiome that they have produces a molecule called 4-EPS. If you inject that into normal pups, they'll develop many of the same symptoms. And you can reverse it with Bacteroides fragilis, which is a probiotic microbe that you can isolate out of the human gastrointestinal tract. So there's a lot of very exciting work going on in these animal models about how you can change the microbiome to change brain conditions. And what we're excited about is translating those into humans. And one thing that inspires me, and this is something John Sanders, one of my postdocs, put together for the EarthMicrobiome Project. A lot of what we're doing right now is reading off the name tags of the bacteria with this thing called 16S ribosomal RNA sequencing. But what we can do is we can get towards the complete genome through further advances in technology that are just coming online now. And so what we're doing is we're moving from nicknames for all of these microbes to being able to get a description of the full name of each of those microbes at the whole genome level, those millions of bases of DNA. And so as we're getting the same sort of growth that we've seen in the EarthMicrobiome Project for sequence tag data and moving that towards whole genome data, you might think, how much sequence data would you need in order to be able to do this? And if we think about every single microbe on Earth, there's about 10 to the 10 species, according to the latest estimates. Each of those has a genome that's about 1 million bases. And if you multiply those together, you get about 10 to the 16 bytes of storage. And to fill that storage with that microbial sequencing would cost you about $30 billion today. But the cost of that sequencing is dropping exponentially, and that scale of data is already in play in a lot of other enterprises. So for example, if you compare it to Facebook, which processes about 10 to the 14 bytes per day and stores about 10 to the 17 bytes, that's 10 times what it would take to store the complete genome of every microbe on Earth, and then use that as a basis for all kinds of functional studies like metatranscriptomics, metaproteomics, metabolomics, that are difficult to interpret today, but with this universal catalog of the microbes would be much easier and much more actionable in the clinic. So we're starting to get towards this idea of a Facebook of life, as it were, and that's exactly the kind of thing we're trying to put together at the Center for Microbiome Innovation that I direct at the UC San Diego, where essentially at this point, with an expanding range of corporate partners and over 120 UCSD faculty members, we're bringing together everyone from ecologists to engineers, everyone across campus, in this collaborative environment that lets us truly revolutionize both how we read out the microbiome and future applications for it, including throughout medicine. So thank you for the opportunity to connect with you, and I think we're answering questions at the panel. Thank you.
Larry and Rob, thank you so much. Now, I'm going to ask a quick question to each of you, but we all want to know, what don't you eat?
Well, aside from processed foods, which there's a tremendous amount of evidence shaping up about how everything from artificial sweeteners to nitrates have deleterious effects on your microbiome and your physiology, I try to cut out useless carbohydrates, so things like bread, rice, potatoes, and so on.
No pizza?
Well, occasionally pizza. I do have a 6-year-old.
He does have a lot of grad students and undergrads. You know.
There you go. Larry.
Yeah, so I've been gluten-free since 2011, and I just did a 85-day experiment of freezing my stool every day and taking blood samples, switching from an omnivore gluten-free diet to a vegan gluten-free diet, and then we'll be measuring later this year and early next year the changes in the microbiome that induced. I have to say that as I did that, the bugs through the gut-brain axis, I pretty much lost interest in eating meat. And the bugs, are really running you. I mean, after all, there's 90% of the DNA-bearing cells in your body are the microbes, not the human. And so it actually turns out that these behavioral modifications include what you desire to eat. And as you changed your diet, the bugs say, I want more of the vegetables and I want less of the other, and you do adapt.
Needless to say, if Larry invites you over to a party at his house, don't go in the freezer looking for ice. Thank you so much, Rob, Larry. Terrific.